Hello everyone, welcome back to our YouTube channel Agri Addict. I am your mentor Ariprasad SK, a PhD scholar from Indian Agriculture Research Institute, New Delhi. In today's video, we are going to discuss about the numerical questions for the ICAR PG exams. So this is very much important for all the students who are going to give their exams in the subject of plant science. So in the last year, I have already uploaded 4 videos which are related to the numerical questions. So out of 4 videos, in first 2 videos there was a problem of audio, so I decided to reshoot the video and I will be uploading it now. So once after the upload, just have a quick look at the questions that you have missed in the previous video, and if you have any doubts you can comment on this video. We will definitely try our best to answer your questions. Okay, without making any delay, let's get started. So very first question is, how many number of primary trisomics can be developed in the maize? So to answer these questions, you have to know two concepts. One is you have to know the chromosome number of maize. So in the diploid state, it is 2n is equal to 20. Okay, and the next thing what you have to know is you have to know what are these trisomics in the first case. So the trisomics is a condition which has a extra chromosomal complement along with the normal complement. That means if the 2n is a normal state of a diploid, along with it there will be one extra chromosomes. So that is what trisomy means. And within the trisomy there are again 3 different classes. So what are they called? They are called as primary trisomics, secondary trisomics and tertiary trisomics. And now they are asking like how many different types of primary trisomics can be developed in the maize which has a diploid number of chromosome number of 20, 2n is equal to 20. So this is their question. So to answer this question, what are these primary trisomics, what are secondary trisomics, and what are tertiary trisomics? So this is very much important. So just observe this by the figure. See, I told you the trisomic is a condition where there is an extra chromosomal complement. For example, let us assume that 2n is equal to 4, right? So that means there will be 2 copies of each chromosomes. So this gives us a representation of 2n is equal to 4. So according to the trisomic condition, if I write it as 2n plus 1 for this 4 chromosome, that means if this is a chromosome number 1 and this is a chromosome number 2, in any of these two there will be an extra complement that will be there with the normal complement. Either the chromosome 1 can be repeated or it can be a chromosome 2 which can be of in the 3 copies. So this condition is called as a trisomic condition. So within this I told you there are 3 different types of trisomic conditions, which are primary trisomics, secondary trisomics and tertiary trisomics. What is the difference between these three? See, in the primary trisomics, the extra complement which you are seeing here is exactly the same of the haploid content. So the same chromosome complement of the normal chromosomes are duplicated, and this is how the extra chromosome complement is formed. So this is exactly same to that of normal chromosome complement. As you can see, this, if we can name it as 1 and this as 2, so same exactly it is matching with the normal chromosome complement. If this is the case, it is called as primary trisomics. In the secondary trisomics, you can see the the chromosomal segment of second part is duplicated on both the arms. Like see, in the normal chromosome complement it is 1 and 2. That means first half is chromosome complement is named as 1 and this is 2. See, 1 and 2 means that they are different. To show that the chromosome arms are different, they are represented as 1 and 2. There is nothing else. Okay, but the extra chromosome which you are seeing here, it has a second arm like chromosome region of 2 that is duplicated. So this is possible because of, if this is the chromosome during the meiosis event, that division, how it occurs, the centromere divides in this fashion. But if in some case if it divides in a vertical fashion or the transverse fashion, you can see that 1 and 1 will be together and 2 and 2 will be together, and this is how the secondary trisomics are formed, where the extra chromosomal complement has identical arms. So this is secondary trisomics. And in the tertiary trisomics, it is because of a translocation of a different chromosomes, translocation of a chromosomal segment from a different chromosomes. Okay, see, the part which is present here, it is not a part of a normal chromosomal complement. If this is the chromosome number 1, this fourth, the part which is been translocated, it is not the region of the normal chromosomal complement. It is the region of a chromosome number 2. Here you can see this arm is named as 4. So some part of this region has translocated to the other chromosome. So this is what a tertiary trisomic means. That means the extra chromosome which you are seeing won't be the same as a normal chromosome complement of that chromosome. It will be similar to that of the other chromosome, where only the part of the chromosome is been translocated. This occurs in case of tertiary trisomics. So now the question is, like, how many number of primary trisomics are possible? As I told you, this extra chromosome is same as a normal chromosomal complement. So it would be is equal to n only. Why? Because the extra chromosome which you are seeing should be the same as the original chromosomal complement. If the 2n is equal to 20 and n is equal to in the haploid set 10, that means there are 10 unique sets, and that will be same homologous to each other. If each has a two homologous chromosomes, that means they are same, right? So this is how we name it as diploid and haploid. In the diploid state, it is just a two identical copies of a chromosomes. So how many unique chromosomes are there within this? If the 2n is equal to 20, how many unique chromosomes you can see? You can see 10 unique chromosomes. If that is the case, the extra chromosomes which will be coming to any of these chromosomes can be of 1, chromosome number 1, or it can be chromosome number 2 or 2, 3 or 4 till 10. So how many different types of primary trisomics that will be possible? It will be always equal to n. This is in case of primary trisomics. But what about secondary trisomics? How many different types of secondary trisomics are possible? So this is 2n now. Why 2n? Because see, here the duplicated part is second 2 and 2 arms, or it can be also of 1 and 1 arms, right? So for the every chromosomes there are two possibilities. So for the 10 unique chromosomes, how many possibilities? Again there will be 20 possibilities. That is why it is 2 into n where it is 10. So we can substitute and the answer would be 20, and this is the case of secondary trisomics. And lastly, it is tertiary trisomics. In case of tertiary trisomics, any part of the other chromosomes can come and place in the extra chromosomal complement, but in this case there are n number of n, not can't name it as n, it might confuse you. There might be as many as chromosomal complements that would be extra are possible. So if they are asking for how many possible tertiary trisomics are possible with the chromosome complement, if they are given the crop or the organisms or directly the chromosome number, then it would be like if the 2n is equal to 20 or any number, it would be like how many number of primary trisomics possible. So it would be n. Number of secondary trisomic possible, it would be 2 into n. And how many tertiary trisomic possible, it would be many. So this is the answer, and you just remember this complete list so that it would be helpful for you if they are asking either primary, secondary or tertiary in the exam. So based on this, our answer will be 10. I hope this is very much clear to all of you. Other than this, there is one more concept that you have to understand, or if you have to know in case of trisomics. So this is about the in the stage of meiosis, how the chromosomes complements are arranged. The arrangement of chromosomes during the meiosis event, how they are arranged. So in the first case, in the primary trisomics, the chromosomes are arranged in a trivalent fashion. This is the most common way of arranging the chromosomes during the meiosis event when the pairing occurs. So if you know the concept, you'll be able to understand this. If you do not know the concept for now, it's okay. You just remember what I'm saying now. If it is asked in the exam, it will definitely help you to answer it. Okay. So what I'm saying is, in the primary trisomics conditions, while the gametes are formed, how they will pair? They will form a trivalent usually. Okay. And in the secondary trisomics, they are forming a bivalent and a univalent. Yes, all other arrangements are also possible, but this is the most common arrangement which I am saying. If in the questions they are asking the bivalence and the univalent formation is possible in case of which type of trisomics, it is only in the case of secondary trisomics. Okay, and if they are asking the trivalent formation takes place in which type of arrangement, whether it is in the primary, secondary, or tertiary, it is in the primary. And lastly, we have this tertiary trisomics. Only in the tertiary trisomics you will get to see the dumbbell shaped arrangement during the chromosomes pairing, or during the gamete formation, or during the meiosis. The dumbbell shape is possible only in the tertiary trisomics. So this is the one more type of questions that you can expect in your exam. Okay, I hope this is clear. Next question: if a plant has a somatic chromosome number of 2n is equal to 14, how many types of secondary trisomics can be developed? So this is the same one which I have explained in the previous slide. So I would be expecting the answers from you. Just comment your answers on the video. I will definitely check it and reply your answers. Okay. Next one. So in the tetrasomic, one chromosome of the complement is present in how many copies? So this is bit tricky. First of all, what is tetrasomic? Tetrasomic is a condition in which a chromosome is present in two extra copies. For example, if 2n is equal to 4, the normal chromosome complement where the 2n is equal to 4, if we arrange it, it would be chromosome number 1 and it would be chromosome number 2. Okay, so in the tetrasomic conditions, either chromosome 1 will have two extra copies or chromosome 2 will have two extra copies. Chromosome number 1, chromosome number 2, either the chromosome number 1 will have two extra copies or the chromosome number 2 will have two extra copies. In both the cases, they are asking one chromosome complement is present in how many copies. So students will definitely get confused here. They just see that, okay, there are two extra chromosomes and 2 will be the answer. No, you have to consider all the chromosomes in the chromosome number 1. That means 1, 2, 3, 4. There will be 4 copies of a one chromosome complement, a single chromosome complement, that will be present in the tetrasomic conditions. I hope this is clear now. Next one: how many different types of gametes are produced by the individual having the genotype capital A small a, capital B small b, capital C, capital D small d, and capital E small e? So seeing these questions, you need not to be worried, since they are given for 5 genes. So there is a direct formula to know the different types of gametes that are formed. The formula is 2 power n. Just remember, to find out the frequency of the gametes or the number of gametes, number of different gametes, the formula is 2 to the power of n. So what is n here? N is, this is very important, the meaning of n. N is not number of genes, it is number of segregating genes. That means, in the simple words, these are number of heterozygous genes. So you have to count the heterozygous genes in the following. This is the heterozygous 1, 2 is heterozygous, 3 is homozygous, 4 is hetero, and 5 is also hetero. There are total of 4 heterozygous genes or segregating genes. So 2 to the power of 4. So what would be your answer? It is 2 into 2 into 2 into 2. It is 4 into 4. That is 16. So 16 will be your answer. So this is how you can solve this type of questions. Okay, next question. A individual has the 4 heterozygous loci where n is equal to 4. This is what they are saying, and they are asking what will be the total number of different genotypes that are produced in the F2. In the previous question, they asked about different types of different gametes, that would be 2 power n. Now they are asking different genotypes. So I'll give you the list of the formulas first. Just try to remember this and revise them before going to the exam. So if there is any question that is asked based on this, you will be easily able to answer it. So what are the formulas? The first point, different kinds of, see, the keyword different kinds is very much important. The different kinds of gametes that are possible in the F1 heterozygous, it will be always equal to 2 power n, that I have discussed in the previous slide. Next, the number of different F2 phenotypes that are possible would be 2 power n if it is for a dominant gene. If there is a single gene which a dominant and recessive behavior, where one gene is showing the dominance and other gene is showing the recessive behavior, in that case the formula would be 2 power n. But if it is for the codominant or the incomplete dominant, then the formula changes to 3 power n. Why so? Let us take an example. Let us assume a seed color we are looking for a seed color, and we are crossing the two different parents, capital R, small r, where capital R is giving red color and small r is giving white color. In this case, the F1 would be capital R, small r, and if we self it, what would be the F2? What are the different genotypic combination? Capital R, small r two times, and small r, small r. These are the four different types of combinations, or four minimum possible combinations that are possible if we are selfing a single heterozygous gene, right? So they are asking us the phenotype. So in the normal case, if it was a completely dominant gene, if R was completely dominant, then we are getting red and again two red and a single white. And what would be the ratio? It is 3:1. The phenotypic ratio would be 3 red is to 1 white. This is in case of a dominant conditions. But if it was co-dominant or incomplete dominant, in both the cases, the heterozygote will give a different phenotype other than the homozygote. Either it might be of any color based on the character. Let us assume it as pink. So here you are seeing that the phenotypic ratio is also divided into three different class. That means one will be red, two will be pink, and other one will be white. So how many different classes you are seeing here? It is 3. So that is why we are considering 3 power n in the codominant or incomplete dominant. But in the earlier case, it was only red is to white. That means two different classes. That is why the formula was 2 power n. I hope this is clear. Okay. Next, the number of different F2 genotypes. But the case is different in case of genotypes. For the genotypes, whether it is dominant, codominant, or incomplete dominant, in all the cases, since the genotype is not changing, even in the incomplete and codominance, you are seeing that the phenotype is changing. Pink, it is converted into pink, but the genotype, it is same. It is same capital R and small r, that is giving the different phenotype. That is why in all the three cases, the formula would be 3 power n. And next, what would be the total number of F2 combinations that are possible? That means what it is trying to say is, if I cross a dihybrid, if I make a dihybrid cross, capital A, capital B, small b, that is self in the F2, what are the possible number of populations that should be present in which all possible crosses should be made? For example, what are the different kind of gametes that are formed? Capital A, capital B, capital A, small a, capital B, and small a, b, right? So this should be combined with every other gamete of the other parent. If we are making every possible gam crosses, then what should be the minimum number of population that we should get? In the dihybrid cross, it is 16. Only if we are able to produce the 16 plants, then there is a possibility that all possible cross combinations can be showed. If the number of the population in the F2 is less than 60, that means any of this possible cross combination is not displayed in the F2. So this is its meaning. So the formula would be the 4 power n. Here in the dihybrid cross, we know at the end of the F2 how many plants we are getting after making all possible crops. It is 16. If we substitute, dihybrid is for the two genes. If we substitute that in this formula, 4 power n is equal to 4 power 2, it would be 16, which is matching. So just remember that the total number of F2 combinations would be, by making all possible cross, would be minimum of 4 power n. It is also called as perfect F2 population. Number of individuals in the perfect F2 population would be 4 power n. Next, the total number of F2 genotypes that are completely homozygous. So for this is 2 power n, where n is equal to number of segregating genes. Next, number of F2 genotypes that are heterozygous. So the homozygous was fine. It is easy for us to remember. For the heterozygous, what we have to do is we have to subtract the 2 power n by the 3 power n. 3 power n is what? It is the total number of combinations that are possible in case of F2. Total number of different genotypes that are possible, or total number of genotypes that are possible, will be 3 power n. And since 3 power n will be total, if we subtract the number of homozygous, what will be left? We are left with only the heterozygous. That is why the formula 3 power n minus 2 power n. Okay. I hope these formulas are very much clear to you all of you. So you can definitely expect some questions, or at least one questions based on this formula. So try to just remember these formulas. So coming back to the question, so it was asking for the different genotypes. The formula is what? The formula is 3 power n. So if we substitute, it is 3 power 4. 3 power 4 is 3 into 3 into 3 into 3. It is 9 into 9. It would be, answer would be 81. Next question. In the case of quantitative trait, which fraction of F2 will be the as extreme as either parent if four pairs of alleles were segregating and there is no dominance? So what they are trying to say is, they're asking you what fraction of the complete population will be similar to the two of the parents. Two of the parents. For example, if I am crossing two different parents, let us assume it as red into white. Okay, so this is the quantitative trait. Since a single phenotype will be governed by more than one gene, here it is the four different genes which this trait is governed, and there is no dominance at all. If there is no dominance, there will be array of phenotypes that we will be getting, in which the extreme would be red and white, which will be similar to that of parent, and intermediate there will be of different colors. So this is with regard to the quantitative trait. And in the when the four pairs are alleles are segregating, so how many of them will be of extreme parent? This is what they are asking. See, first of all, what we can do is we can construct a perfect F2 population. So what will be the size of perfect F2 population? I told you it is 4 power n. It would be 4 to the power of 4, so which comes up to be 256. So we have to calculate out of 256 how many of them will be red and white. So this is their question. If you consider the dihybrid cross, you can clearly see only the extreme, either of the extremes at the table will be of parental type. That means it is dark red and white. So how we are getting dark red? It is by the combination of having all dominant genes together. And how we are getting white? It is with the small r, which is for 4 times. This is how we are getting a white. That means it is completely homozygous recessive, and the other one is completely homozygous dominant. Out of this 16 different combination, in case of dihybrid cross, we are getting only two fractions, that is 2 by 16, that would be similar to that of parents. Similarly, even if you go for a tetrahybrid cross, then even then the fraction will be 2 by 256 only. Out of 2, out of 256, there will be only two combination which will be completely homozygous for either dominant gene or for the recessive gene. For this, the answer would be 2 by 256, since they have asked for either of parents. Either means or, that means we have to add the individual frequency. That means 1 by 256, 1 by 256, it would be 2 by 256, not 1 by 256. Okay, I hope this is clear. Next, this is a question in which they are asking, there is an incomplete dominance and how many different phenotypes will be observed in F2 if there are 4 pairs of alleles that are segregating. That means again n is equal to 4, and they are asking for a total number of different phenotypes if there is a incomplete dominance. In this case, I told you the formula changes from 2 power n to 3 power n. So now we have to substitute n value for 3. 3 into 3 into 3 into 3. It would be again 81. I hope this is also very much clear to all of you. Next one. So this is the probability question. So normal girl is married to normal male, and out of their 4 children, one child has a recessive genetic disorder. That means the genotypic combination of this particular children should be of small a, right? And next, it can be of any gene. Like I am assuming that it has, like, gene would be A. If gene is A, dominant phenotype and the heterozygous phenotype will be giving the normal, and the recessive homozygous will be giving the diseased phenotype, right? So that is why I'm considering it as capital A, capital. You can consider any alphabet you want. Next, what are the genotypes of the two parents for the gene A, which is responsible for this disease? They are asking to go for its parents if in the progeny there is a child out of one, like 1 out of 4 children's who is, or he or she is having the genetic disorder. So it's very simple to understand that if we want in the progeny, if we want to get capital A small a, sorry, small a as a combination, we should be having this allele in both the parents. It should be present in the both the parents, at least in a one copy. It should be compulsorily should be present in both the parents if it is female and male in a single copy. But the other copy, what it can be, that we can see here. So to answer this, simply we can just try all the cross combination. In the first combination, the parent is considered as capital A, capital A, capital A, small a. As I told you, the parent should at least have one small a. In the first case, there is no small a at all in the female. So this option is wrong. In the second case, it is capital A small a and small a small a. In this case, both the parents are having small a, but if we cross them, the gametes are like capital A small a, this is the genotype, and the small a small a. There are only two different genotypes are formed, which is equal to 1:1. But they are saying that out of 4 children, only one is recessive. All three are normal. One is only diseased. So this combination is not possible with this parent. This is also wrong. And the third one, as you can see, again there is no small a recessive allele in the female parent. So this is also wrong. Let us try the fourth one. In the capital A small a, capital A small a, so we know that it is capital A, capital A, capital A small a, capital A small a, and small a, out of which this is 3 times and this is the 1. That is, phenotypic ratio would be 3:1. 3 normal and 1 would be diseased. So this is exactly matching to our questions, out of which out of 4 only 1 is diseased. So this will be our answer. Next one. The F2 generation from a cross between albino and the black mice produced 75:33 albino progenies. Find out the correct expected ratio from the among the following options. So seeing the options, as you can assume, as we can clearly know that it is for a single gene and a single trait. The trait is already been given here, that is albino and the black. So this is a code color. Okay, for this the observed is 75:33, but what should be expected here? If it is a single cross, like single gene with a single trait, we know that if we are crossing a single gene for a single trait, then in F2, if we are selfing it, we have to get capital A small a, capital A small a, and small a small a. It should be 3:1. That means 3 of which will be of black strains and 1 will be of albino. So this should be the case. So the answer would be 3:1 here. This is the expected if this is the case. Next, which of the following formula is used to finding out the minimum number of individuals in a perfect F2 if n is the number of segregating generation? As I told you previously, if they are asking for the perfect F2 population which contains all possible cross combinations, it is 4 power n. Remember this, it is 4 power n. Next one, they are asking a family as a 4 children, what is the probability that 2 are girls and 2 are boys? So this is of more of probability questions. You may or may not expect this type of questions in the exams, but since it is very easy to solve, we'll just try to solve it. See, it is with the formula NCR, P power n minus r, and Q power r. And what are P and Q? So this is the probability of sexes. Let us consider one event now. This is probability of getting 2 girls. So whenever the 2 parents are mated, the female with the male, what will be the probability of getting a boy and a girl, or a male and a female? The probability will be always equal to 1/2 and 1/2, whatever might be the generation. For example, if in the first generation, what is the probability of getting boy, and what is the probability of getting a boy in the next mating, what will be probability in the third mating, fourth mating, or the nth mating? The probability will be always same. It would be 1/2 for male and 1/2 for female. That means there is an equal probability of getting either boy and girl, right? So the P will be equal to half, the probability of sexes. And what will be Q? Q is nothing but 1 minus P. It will be again equal to half. And what is R here? Since we are considering a single event now, one event, which is probability of getting 2 girls, so that would be the R value is equal to 2. If you substitute, it is n value is equal to 4 here, since the total number of children are 4. 4CR, 4C2 into P power 1/2 into 4 minus 2 into 1/2 into 2. So this gives us 4 into 3 divided by 2 factorial into 2 and divided by 2 into 1/2 into to the power of 2 into 2. So this will be giving us the answer of 6 by 16. If we solve this, the answer would be 6 by 16. Next one. What will be the frequency of heterozygous A small a in the random mating population in which the frequency of the dominant phenotype is 0.36? Just by seeing these questions, you might be confused like how it can be solved, but this is very easy to solve since they have clearly given that it is a random mating population. If it is a random mating population, we can assume that it is in the Hardy-Weinberg equilibrium. If that is the case, we know the formula equation that p square plus 2pq plus q square is equal to 1. So what they have given here, they have clearly given the value of p square plus 2pq, that is capital A frequency of capital A plus frequency of capital A small a plus q square is equal to 1. So if we substitute and try to find out the answer, it is 1 minus 36, which would be q square would be 0.64 and q would be 0.8. So this is the value of q, and what will be the value of p? The p would be again 1 minus q, it would be 0.2. So this is the value. And then what they are asking the frequency of heterozygous. The heterozygous, we know that it is 2 into pq. So 2 into p is 0.2 and q is 0.8. If we substitute, it is 16 to the 32, it is 0.32. So answer would be 0.32. Okay, next one. If the observed data subjected to chi-square test has a 6 classes and total of 1000 observations, what will be the following, which of the following will be the degrees of freedom of a estimated x? So for this one to answer, we have to know what is this degrees of freedom. I guess you might be already knowing the direct formula for the degrees of freedom, which is n minus 1, and n will be equal to the total number of classes. That's why it would be equal to 5. So this is true. But what exactly this degrees of freedom means? So to understand this, let us take an example. Let us consider there is a box in which there are 4 different balls of different color: black, red, green and orange. And there are 4 students, or the 4 children, they are standing in a line, 1 2 3 4 4. They are standing in a queue, and each of these students or the children are allowed to pick one ball from the box one by one. So now just assume, like imagine that the first person or the first child just comes to the box and just look at the box and try to take one ball. So how many options do he has? Like when child comes and see the colors, he has a 4 options. That means first child will have a 4 options. Either he can choose one color ball with any of these 4 colors. So he will just come and pick the red, let us assume. So the red is gone. The second child will come to the box and see, and how many options does he have? He has 3 options, and let us assume he will choose the brown color, and this is gone. The third child will come and he will look at the box, and how many options he will have? He will have 2 options. He will pick any of that color. Let us assume it is picking green color. Now the fourth child, if he comes to the box, just see, like if he has any option, he has to pick the orange ball only. He has to pick the orange ball only, and he has no option. So this is what the degrees of freedom is, how much freedom is given for the selection. So in this case, if there are 5 class, for selecting the 5 classes there is option or the freedom, but selecting the 6th class there is no option. That is why the degrees of freedom will be equal to only 5. It will be, that is why the formula n minus 1, and here the answer would be 5. I hope this is clear to all of you. Next one. This is a simple question which they are asking. A family has a 4 girls. So what is the probability that the 5th baby born in this family will also be girl? So this is just to trick you. Even if it has a 4 girls, 5 girls, 4 boys, 2 boys and 2 girls, or whatever might be the combination, if this is the independent events, that means the second event is not influenced by the first event, then the probability always remains the same. So in the 5th case also, the 5th boy in the family is also be girl. I told you whether it is boy or girl, the probabilities will be 1 by 2 and 1 by 2. So the answer would be 1 by 2 here. So you need not to get confused with this. Next one. Under optimum conditions, one complete replication of E. coli takes approximately how much time? So this is also a bit tricky, where students will directly mark it as 20 minutes, since the complete generation time of E. coli is 20 minutes. But the question, they are not asking about the generation time, they are asking about the replication time. In the E. coli, the replication time is actually 40 minutes, not the 20 minutes. It's approximately 40 minutes. Okay, so the 40 would be your should be your answer. Again, seeing this, you might again get one good question. What is it? If in case the replication time is itself 40, and how the E. coli is able to complete its life cycle in just 20 minutes, how it is possible? So this is because of the presence of special type of replication which is called as dichotomous replication, in which the replication, before the finish of first replication, there will be a second set of replication that is already started. This is how they compensate the time, and the generation time will be over within 20 minutes. Okay, fine. Next one. If the q is equal to 0.2, what will be the frequency of capital A, small a, and will be which of the following? So if the q is equal to 0.2, what will be the frequency of genotype capital A, sorry, small a, small a? So this is also very much simple. Since q is equal to 0.2, we know that q square will be is equal to frequency of small a, which will be equal to 0.2 into 0.2. That would be equal to 0.4, 0.04, sorry. Next one. If the a is equal to q, q is equal to 9.9, and small a is equal to 1 minus q, that will be 0.1. What will be the frequency of three genotypes, which of the following? So they are asking the p square, 2pq, and q square values. You just substitute these p values and get the answer, and comment your answer in the video. Next one. So next question, it is, in the natural population of naturally self-pollinated crops, the value of coefficient of inbreeding, which is represented as F, will be of which of the following? For this question to answer, we have to know the concept of coefficient of inbreeding, which is represented as F. What is F? F is a probability of two alleles, which are identical by descent. That means what will be the probability that the two alleles have been transferred from the same individual. So if we this clearly see this pedigree, we'll be able to answer it. So in this case, the capital A, small a, capital A, small a. So different colors are to denote that or represent that they are from which parents. In this case, as a generation goes on, you can see the A is being go on like transferred to this individual, and A is been transferred to this individual. When these sisters exhibit mating is done, brother sister mating, you can see that the capital A and capital A have been come into a single individual, which was actually present in a same or a single individual. The allele that was present in a single individual, that came into a individual in its progeny generation. That means this is the probability of two alleles which are identical by descent. This is its meaning. Okay, and they are asking now, what will be the value of inbreeding, like coefficient of inbreeding, which of the following? In this case, remember that if it is completely self-pollinated crops, it will be always equal to 1. The f value will be equal to 1, and the f value, it would be between 0 to 1. If it is 1, that means it is a complete inbreed population or self-pollinated population, or if it is 0, then it is completely a random mating population. So just try to remember these two things. This might directly come in your exams. F value equal to 1, it is an inbreed population. F value equal to 0, it is a random mating population. Okay, so that's it for this video. In the next video, I'll be discussing the part 2 of these numerical sessions. If you like our content, please like, comment, share, and subscribe our YouTube channel, and also do answer whatever questions I have asked in this video. I will be definitely looking at the comments, and I will be replying to your answers. Okay, thank you so much.